Welcome to the PubMed Daily Review for March 17th. Today we scan 17 new open access papers. Here are the top 5 selected for detailed analysis. Trend 1 of 5, Surgical Oncology and Treatment Strategies. These papers explore advancements and best practices in surgical approaches and outcomes for various cancer types. This cluster covers 5 papers. Representative work includes the prognostic significance of elective level 4 neck dissection in oral tongue cancer, a systematic mapping review of core outcome reporting in surgical research for esophageal cancer, and Brazilian Society of Surgical Oncology, guidelines and consensus statement for palliative surgery in oncology. Trend 2 of 5, Metabolic and organ-specific outcomes. Investigating how specific organs and their metabolic functions impact overall health and disease. This cluster covers 3 papers. Representative work includes long-term metabolic outcomes following pancreatectomy and autologous islet transplantation, systematic review and meta-analysis. Cytoarchitectural multidepot profiling reveals immune-metabolic crosstalk in human colon-associated adipose tissue, and cardiac output measurement in Malawian children ages 2 months 12 years hospitalized with severe anemia. ComTract. Trend 3 of 5, Cancer Metastasis and Molecular Mechanisms. These papers explore how cancer spreads, focusing on blood vessel involvement and molecular signaling pathways. This cluster covers 2 papers. Representative work includes vascular reconstruction in extremity soft tissue sarcomas, a systematic review and single-arm meta-analysis, and aspartate signaling drives lung metastasis via alternative translation. Trend 4 of 5, Respiratory Health and Environmental Impact. These papers explore how environmental factors and physiological conditions impact respiratory health and related developmental outcomes. This cluster covers 3 papers. Representative work includes application of a human bronchoepithelial air-liquid interface model to assess respiratory hazard of VOCs using a benchmark concentration modeling approach, respiratory inductance plethysmography to quantify changes in ventilation in obstructive sleep apnea, and maternal infection but not inflammation is associated with attention deficit/hyperactivity disorder symptoms during childhood, a population-based cohort study. Trend 5 of 5, Fundamental Biological Processes and Health Disparities. This trend examines how fundamental biological processes intersect with health disparities across diverse organisms and populations. This cluster covers 4 papers. Representative work includes disparities in breast cancer screening, diagnosis, and outcomes among Vietnamese American women, a systematic review. The function of the Arabidopsis receptor kinase theseus-1 in plant cell wall integrity maintenance, from evolutionary origin to future perspectives, and how growth-induced stresses guide shape changes during animal morphogenesis, mechanisms and implications. Up next, our first paper out of 5, Aspartate Signaling Drives Lung Metastasis via Alternative Translation by Ginevra Doglioni and Sarah Maria Fent from Laboratory of Cellular Metabolism and Metabolic Regulation, Center for Cancer Biology, Herestrat 49, 3000, Leuven, Belgium. Let's break down its key contributions. Lung metastases are a major challenge, affecting a significant portion of patients with advanced cancers. The lung's unique environment and the factors it provides are thought to help cancer cells survive and thrive. However, we still have much to learn about how specific nutrients available in the lung might actually make these metastatic cells more aggressive. To investigate this, the researchers looked at breast cancer models in mice and human patient samples. They focused on the role of aspartate, a nutrient found in high concentrations in the lungs of affected patients. Using various molecular techniques, they traced a signaling cascade initiated by aspartate that ultimately enhances the aggressiveness of lung metastasized cancer cells. The study found significantly high levels of aspartate in the lung interstitial fluid of patients and mice with breast cancer. This extracellular aspartate was shown to activate the N-methyldiaspartate receptor on cancer cells. This activation initiated a cascade, leading to increased expression of DOHH, which is crucial for a process called hypocyanation, ultimately boosting the aggressiveness of the cancer cells through enhanced collagen synthesis. This research reveals a novel role for aspartate, not just as a building block, but as a signaling molecule that promotes lung metastasis. The findings highlight a specific pathway involving NMDAR activation and subsequent translational changes that enhance cancer cell aggression. This opens up exciting possibilities for developing new therapies aimed at blocking this aspartate-driven mechanism to combat lung metastases. Up next, our second paper out of 5, Cytoarchitectural multidepot profiling reveals immune-metabolic crosstalk in human colon-associated adipose tissue by Jutta Jokinen and Niklas Medjert from Department of Medicine, H7, Karolinska Institutet, C2-94, Karolinska University Hospital, Stockholm, Sweden. Let's break down its key contributions. We know that fat tissue isn't all the same. Different depots, like the ones under our skin versus those around our organs, have different cell types. But what does this actually mean for how these tissues work? This study aimed to map out these cellular differences and uncover their functional significance. To tackle this, the researchers used advanced techniques, combining spatial mapping with single-nucleus RNA sequencing. They analyzed various fat depots from both men and women to create a detailed cellular map. This allowed them to identify unique cell populations and communication pathways specific to each depot, particularly highlighting the colon-associated adipose tissue. The study found that fat tissue near the colon, specifically the epiploic depot, was unique. It contained a high number of fat cells expressing SAA1 and SAA2. Importantly, these fat cells ramped up SAA1 production when exposed to inflammatory signals, and this SAA1 then triggered immune responses in nearby immune cells. This shows a clear immune-metabolic crosstalk. So, what does this mean? It highlights that fat tissue isn't just a passive energy store, it actively communicates with the immune system, especially in visceral depots. This specialized crosstalk, particularly involving SAA1, could be a new area to explore for treating inflammatory diseases. Future studies will likely delve deeper into these interactions. Up next, our third paper out of 5, maternal infection but not inflammation is associated with attention deficit/hyperactivity disorder symptoms during childhood. A population-based cohort study by Milan Zarchev and Veerle Bergink from a Department of Child and Adolescent Psychiatry/Psychology, Erasmus MC, Rotterdam, the Netherlands. Let's break down its key contributions. We know that ADHD is a common neurodevelopmental disorder, but what causes it? Researchers have long wondered if things happening during pregnancy, like infections or inflammation, could play a role. This study aimed to clarify whether maternal fever, infection, or inflammatory markers are truly associated with a higher risk of ADHD symptoms in children. To investigate this, researchers followed over 6,500 mother-child pairs from birth. They collected data on maternal infections, fever, and specific inflammatory markers like cytokines and CRP during pregnancy. The children's ADHD symptoms were then assessed using parent and teacher reports at ages 6, 10, and 14, allowing for a comprehensive look at the association over time. The results showed a small, yet statistically significant, association between maternal fever during pregnancy and higher ADHD symptoms in children. However, markers of inflammation like cytokines and CRP did not show any significant link to ADHD symptoms. Furthermore, the study found no evidence that genetic predisposition or maternal stress during pregnancy modified these associations. So, what does this mean? While the effect size was small, maternal fever during pregnancy is linked to offspring ADHD symptoms. This highlights the importance of managing fever during pregnancy. Interestingly, chronic inflammation markers didn't show a link, suggesting a more specific mechanism at play. Future studies should explore why fever, specifically, might influence ADHD development. Up next, our fourth paper out of 5, application of a human bronchoepithelial air liquid interface model to assess respiratory hazard of VOCs using a benchmark concentration modeling approach by Olivia C. G. Lampe and Natalie M. Johnson from A Department of Environmental and Occupational Health, Texas A&M University, College Station, Texas, USA. Let's break down its key contributions. Volatile organic compounds, or VOCs, are everywhere, in our homes and outside. We know they can cause health problems, but how exactly do they affect our lungs? This research uses a special lab-grown model of the human airway to find out. Researchers used a sophisticated in vitro model, growing human airway cells at an air-liquid interface. These cells were exposed to two common VOCs, acrolein and formic acid, or clean air. They then measured cell damage, how well the airway barrier held up, and the activity of genes related to inflammation and stress. The study found that both acrolein and formic acid caused significant cell damage and made the airway barrier more permeable, especially at higher concentrations. They also triggered genes linked to inflammation and oxidative stress. Formic acid specifically increased a gene involved in cellular cleanup called mitophagy. This research shows that our lab-grown airway model is a promising tool for understanding how VOCs affect our lungs. It confirms that these chemicals can indeed harm airway cells. Ultimately, this work could lead to better safety standards and protect us from harmful airborne compounds. Up next, our fifth paper out of 5, long-term metabolic outcomes following pancreatectomy and autologous islet transplantation. Systematic review and meta-analysis by Daniel L. Hughes and Giovanni Marchigiani from Department of Hepatopancreatobiliary Surgery, Oxford University Hospitals NHS Foundation Trust, Oxford. Let's break down its key contributions. Total pancreatectomy, the removal of the entire pancreas, often results in severe diabetes due to the loss of insulin-producing cells. Autologous islet transplantation, where a patient's own islets are transplanted back, is a procedure aimed at mitigating this. But how well does this approach work in the long run? This review sought to answer that very question. To understand the long-term effects, researchers conducted a systematic review and meta-analysis. They analyzed data from 17 studies, encompassing a total of 1,332 patients who underwent total pancreatectomy with autologous islet transplantation. The primary focus was on metabolic outcomes, particularly the rate of insulin independence over extended periods. The analysis revealed a pooled insulin independence rate of 34% across all patients. Importantly, this rate was substantially higher, at 68%, for those whose TPIAT was not indicated for chronic pancreatitis, compared to 33% for those with chronic pancreatitis. Overall, the findings suggest that TPIAT is effective in preserving endocrine function. In conclusion, this meta-analysis highlights its effectiveness in preserving endocrine function, especially when chronic pancreatitis isn't the primary indication. However, there's a clear need for more standardized reporting, particularly concerning long-term metabolic health and patient-reported quality of life. Future research should aim to validate these findings across a broader range of patient groups and indications.